Hello, I welcome you all to today's session where we are going to discuss the next part of the biomolecules, that is the enzymes. So, enzymes are those substances which catalyze various reactions. A spontaneous chemical reaction can occur without any requirement of outside energy, but it can occur so slowly that it is practically negligible for the living system. So an enzyme is a macromolecule that acts as a catalyst or a chemical agent that speeds up a reaction without being itself consumed in the reaction. This is the definition that we have been studying since our lower classes. Enzymes are usually the most remarkable and highly specialized proteins having an extraordinary catalytic power and a high degree of specificity for their substrates. So enzymes are basically proteins. But there are some RNA molecules which are present inside the cell and they can also act as enzymes. So these RNA molecules which act as enzymes, they are otherwise called as ribozymes. And they can also function just like that of the enzymes. Enzymes are core to every biochemical process as they catalyze the stepwise reactions which are involved in degradation of nutrient molecules or the conservation and transformation of chemical energy as well as synthesis of biological macromolecules from simple precursors. The molecule or the biochemical which is acted upon by the enzyme is otherwise called as a substrate. But when more than one substrates are involved in a chemical reaction, then they are called as the reactants. So once we write the equation for the enzyme catalyzed activity, this concept of substrate and reactants will be more clear to you. The chemicals which are formed after the completion of the reactions, they are otherwise called as the products. So now let us write a simple equation where A plus B are the two chemicals which are reacting in presence of the enzyme to form the chemicals C and D. So I will be designating this A plus B as the reactants and C plus D as the products. Having said this, let us come to that who has coined the term enzyme first. The term enzyme first was coined by Kuhn and it was the year 1877. But he gave this term for catalytically active substances. But the enzymes were first discovered and isolated by Buckner. And that was almost 20 years after the discovery of Kuhn, that is in the year 1897. And for this important discovery, Buckner was awarded Nobel Prize in the year 1903. So since enzymes are capable of catalyzing reactions of biological origin and they are synthesized by living cells only, so sometimes they are designated as biocatalysts. So now our further discussions we will be concentrating on biocatalysts only. Depending upon where the enzymes are acting, that is whether they are acting outside the living cells or they are acting inside the living cells, depending on that, enzymes can be of two types. They can be either exoenzymes or they can be endoenzymes. Suppose this is our curved J-shaped sac of the stomach. So there are some cells which are lining the stomach and these are the cells which secrete the gastric juice and that contains a lot of digestive enzymes. So this area is called as lumen. So if the food material is getting digested in this area, then how do I designate this? I designate this that it is inside the lumen of the stomach but outside the cell. So all your nutritive enzymes or your digestive enzymes, they are exoenzymes only. But now you compare this concept with the enzymes of the Krebs cycle. You know that the Krebs cycle takes place inside the mitochondria of the cell. So where mitochondria is present, it is present inside the cell. So all the enzymes which are involved in Krebs cycle, they will be endoenzymes, as well as the enzymes which take part in the process of glycolysis. Glycolysis takes place in cytoplasm. So whatever takes place inside the cell, whether it is in this area or it is inside the mitochondria, they will be all endoenzymes. Having said this, let us come to that what are the chemical nature of the enzymes. With the exception of a small group of catalytic RNA molecules, that the ribozymes that I have already told you, all enzymes are globular proteins. Remember the primary, tertiary, secondary, quaternary structure of proteins that I have taught you. So they are all globular proteins. Some enzymes may also contain the non-proteinaceous molecules and metal ions that participate directly in the substrate, either binding to it or catalyzing it. Based on the presence or absence of non-protein groups, enzymes can be classified into the following categories. So how do we classify the enzymes? Basically, the enzymes will be classified as follows. They can be simple enzymes or they can be conjugated enzymes. So they can be simple or they can be conjugated. Having said this, let us understand what are the simple and the conjugated enzymes. In case of simple enzymes, they will be made up of only proteins. There will be nothing else in their structure. Secondly, the active site of this enzyme will be consisting of specific groups of its own amino acids. So the active site of the simple enzymes will be made up of amino acids or specific protein chains, and the enzymes which are involved in our digestion process like pepsin, then trypsin, these enzymes will be coming under this category. If you compare this with conjugated enzymes, the enzyme will be consisting of two parts. Each conjugated enzyme will be having a proteinaceous part that is otherwise called as the apoenzyme. So apoenzyme is the proteinaceous part and the non-protein part is otherwise called as a cofactor. So cofactor is the non-protein part of the conjugated enzyme. And the complete enzyme means apoenzyme plus cofactor. This complete enzyme will be known as a holoenzyme, and the more popular members under this category will be having the active site which is partly formed by the apoenzyme and partly formed by the cofactor. Having said this, let us understand what is a cofactor. Cofactor is a small heat stable and dialysable part of the conjugated enzyme, which may be organic or inorganic in nature. It is also dialysable, and cofactor can be organic or it can be inorganic in nature also. Cofactors are of two types. They are otherwise called as the coenzymes, or they are also called as the prosthetic groups. So if I write that what are the further classification of cofactors, then cofactors are further classified into coenzymes and prosthetic group. Coenzymes are easily separable. The first characteristic is that they are easily separable from the rest part of the enzyme. Secondly, they are non-protein. Thirdly, they are organic in nature. Sorry, I think this part is not visible to you. So they are organic in nature, and they act as transient carriers of specific functional groups. They require two apoenzymes, one for picking up the group and the second for transforming the group. For example, your NAD that we have studied during that process of respiration, NADP plus that we have studied during photosynthesis, coenzyme A that we have studied during respiration. These all are coenzymes. Most of the coenzymes are made up of water soluble vitamins like vitamin B and vitamin C, like thiamine, riboflavin, nicotinamide, pyridoxine, etc. If you compare the coenzymes with the prosthetic groups, then prosthetic groups are also non-protein. This is the fact which remains common between the cofactors and prosthetic groups. They are also organic, but they remain very tightly associated. This is the major point of difference between the coenzymes and the prosthetic groups. Prosthetic groups will be very closely associated with the apoenzymes and they take part in group transfer reactions, and the example under this will be heme of hemoglobin, biotin, etc. You will find that inorganic cofactors, the third category, they will be consisting of ions of various minerals, that is calcium, iron, copper, zinc, nickel, molybdenum, etc. They usually function as activators by forming one or more coordination bonds with the substrate and the active site of the enzyme. So cofactors can be of another type, that is inorganic. And when we are talking about the inorganic cofactors, so basically we are talking about this type of inorganic substances. I can give you some examples like copper, it will be a cofactor for cytochrome oxidase. Ferrous or ferric forms, they will be associated with enzymes like cytochrome oxidase, catalase, peroxidase. Potassium ions will be associated with pyruvate kinase. Divalent magnesium ions are obviously associated with hexokinase, glucose 6-phosphatase, pyruvate kinase. Remember these enzymes we have studied when we were studying either glycolysis or Krebs cycle. Manganese, divalent manganese, that is Mn2 plus, that will be associated with, if I am remembering it correctly, the enzyme ribonucleotide reductase. Molybdenum will be associated with dinitrogenase. With urease enzyme, you will have the association of nickel, and selenium will be associated with glutathione peroxidase enzyme. Zinc will be associated with carbonic anhydrase. This we have studied in respiration. Alcohol dehydrogenase will be also having inorganic cofactor of divalent zinc, and carboxypeptidase A and B, which are digestive enzymes, also contain zinc. So having said this, for our further clarity of understanding, let us compare the apoenzyme and the coenzyme. Let us compare the two terms so that we understand it better. Apoenzyme will be the protein part of the holoenzyme or the conjugated enzyme. So it is the protein part. Whereas coenzyme will be purely the non-protein part of the holoenzyme. So coenzyme will be an organic group which gets associated to the apoenzyme to form the holoenzyme or conjugate enzyme. Apoenzyme, as it is made up of protein, so it will be comparatively larger in size. Coenzyme, obviously in comparison to apoenzyme, that will be smaller in size. Apoenzyme is very specific for a particular enzyme. One apoenzyme cannot work for so many enzymes, but that is not the case with coenzyme. A coenzyme can function as a cofactor for a number of enzymes. Like just now, I have given you the example of zinc, though it was a cofactor. Then apoenzyme takes part in catalytic activity of the enzyme. So this will only take part in the catalytic activity. But the coenzyme, as we have discussed, it helps in removing a product of chemical reaction, as well as it will be responsible for bringing the substrate and the enzyme in close contact. So it helps in mostly transfer or contact type of reactions. Then apoenzymes do not help in group transfer. Obviously, this part will not be involved in transfer of anything from one place to another, because it will be mainly associated with catalyzing activities, right? So next, obviously coenzyme will take part in group transfer reactions. Then apoenzyme, that is, it can be destroyed by heat, or it is negatively affected by heat, so it is thermolabile. But this coenzyme part, it is quite heat stable. I am telling that it is quite heat stable. If you apply a more than specific amount of heat, then it can dissociate it from the apoenzyme, right? So now let us come to the active site of the enzyme. This will be another important term, and we will be coming across what is the active site of an enzyme many times. Only a restricted region of the enzyme molecule actually can bind to the substrate, not the entire enzyme. Suppose this is the total enzyme and this is the particular substrate. As you can mark the shape of the substrate, it is not going to bind anywhere. It is going to bind only to this particular slot like structure, which I have written as active site. So this substrate can bind only to this region, not to the entire part of the enzyme. So that V-shaped depression like area that I have drawn, that is the active site. Active site of the enzyme is a small portion of the enzyme that takes part in catalyzing biochemical reaction by attracting and holding a particular substrate. And how it can hold so? Either by its specific charge or by size or by its shape. The active site generally consists of a few amino acids, because remember, except ribozyme, I have already repeatedly told that most of the enzymes are proteinaceous. So if the enzyme is proteinaceous, then its active site, that will be consisting of amino acids. As you can see, if the total shape of the enzyme is like this, then only this V-shaped portion will be consisting of very small number of amino acids. So that active site will be made up of small number of amino acids or their side groups, which are brought together in a particular fashion due to either the secondary or tertiary folding of the protein molecule and its association with the cofactor. If the cofactor is at all present, it is possible that one enzyme can have more than one active site. When we come about the inhibition of the enzymes, this term will be more clear to you. So till now we can discuss, or we can summarize, that an enzyme is not a very stiff structure that confines into a given shape. Recent work by biochemists have showed that the enzyme is said to you can say fluctuate between different shapes in a dynamic equilibrium, and these shapes are slightly different from each other if we consider their free energy. And as the substrate enters the active site, the enzyme changes the shape slightly, because the interaction between the substrate and the chemical groups that are present in the active site region and on the side chains of the amino acids that form the active site. So now let us discuss what are the different classifications of the enzymes. We have already studied their chemical nature, then we have also studied what does its active site mean. Now let us understand that how enzymes are classified. Enzymes are classified according to the modern system of enzyme classification, which is introduced by the International Union of Biochemists, that is IUB, and this is in the year 1961. Enzymes were classified basically into 6 classes based on what type of reactions they catalyze. Recently, the International Union of Biochemistry and Molecular Biology has added a new class of enzymes, that is translocases. As we will discuss, I'll tell this to you. And now it brings it to 6 classes of enzyme classification. So the first class of enzyme that we are going to study, or according to this classification of International Union of Biochemistry and Molecular Biology, the first group is that of a hydrolase enzyme, or popularly which are called as the hydrolases. These enzymes catalyze the splitting of larger molecules into smaller ones by addition of water. Because they hydrolyze the bonds, or they bring about the hydrolysis of bonds, that is why they are called as hydrolases. And that they do so with the help of hydrogen or OH minus, that is hydroxyl groups. Usually the bonds they attack can be the ester bonds, the ether bonds, the glycosidic bonds, which are formed either by dehydration condensation reactions, and the enzymes which work under this particular category can be amylase that digests the starch, sucrase, lactase, etc. So if I write the equation like this, that starch under the influence of the amylase enzyme, we already know it breaks into a disaccharide which is called as maltose. Sometimes it can also yield isomaltose, or it can also produce limit dextrins, or alpha dextrins we call it. So this reaction of breaking of starch into maltose, that is catalyzed by the enzyme amylase, which is a hydrolase. Then the second category that we are supposed to discuss, that is oxidoreductase. The name itself indicates that it might be catalyzing the oxidation reduction reactions. These enzymes are basically of three types: oxidases, they can be oxidase, cytochrome C oxidase, remember this name, this will come under this category. It can be dehydrogenase, succinate dehydrogenase we can put under this category, or they can be reductases. The example under this category will be nitrate reductase. So the reaction that it catalyzes, say for a cytochrome oxidase, that if right, then it is NADH plus H plus half O2 in presence of cytochrome oxidase, that will be changing itself into the coenzyme NAD plus evolution of oxygen. Then the next category of enzymes, they are called as transferases. The name itself indicates that they help in transfer of certain groups from one substrate to another to form the product. They catalyze the transfer of specific groups other than hydrogen from one substrate to another. Otherwise, they will be classified as dehydrogenase. The group transfer does not occur in the free state. So, as your nitrogen fixation is standing deleted from the syllabus, had we been studying that particular part, there you would have learned about the synthesis of amino acids. So, there is a specific reaction which is given in that particular chapter of the NCERT book, where an amino acid glutamic acid reacts with oxaloacetic acid, which is an organic acid, in presence of the enzyme transferase, and that brings about the formation of alpha ketogluteric acid, and this alpha ketogluteric acid is an organic acid, and formation of aspartic acid, which is an amino acid. So I had made this amino acid glutamic acid to react with this organic acid oxaloacetic acid. I have shifted certain groups from oxaloacetic acid to glutamic acid to form again one organic acid, that is alpha ketogluteric acid, and the amino acid aspartic acid. This is under the chapter 12 of your NCERT book. Then the next category of enzymes, they are the lyases. The word lysing means breaking down. So lyases might be catalyzing that kind of reactions. These enzymes catalyze the breakdown of specific covalent bonds and removal of groups without performing hydrolysis, and they produce double bonds or removal of double bonds by adding organic groups. Remember, when during glycolysis, fructose 1,6 bisphosphate or fructose 1,6 diphosphate, which is a 6 carbon containing compound that in presence of aldolase forms 2 3 carbon containing compounds, that is 3 phosphoglyceraldehyde plus dihydroxyacetone phosphate. This is also 3 carbon, this is also 3 carbon, and this is a 6 carbon compound. This particular reaction is catalyzed by the enzyme aldolase, which is a lyase. The next category, that will be that of isomerases. Obviously the name indicates that they will be catalyzing the formation of isomers. These enzymes catalyze the change of a substrate into its related isomeric form by rearrangement of molecules. The enzymes are again further classified into 3 types, that is isomerase particular A. In this case, they change aldose into ketose or vice versa. Proper isomerases change aldoses to ketoses or back ketoses to aldoses. The second group is that of epimerases. If you remember the isomerism that we have discussed under carbohydrates, you will remember this. Epimerases generally will be changing the position of one constituent or carbon group into other. The third category under this will be that of mutases. So these particular enzymes will be shifting the position of a side group like glucose 6 phosphate to glucose 1 phosphate. The phosphate group which is present at carbon atom number 6, that will be shifting to carbon atom number 1. The next group of enzymes, that will be ligases, means they join something, ligate something. These enzymes catalyze the joining of 2 substrate molecules by utilizing the energy by hydrolyzing ATP. So we can write this famous reaction, that is pyruvate plus carbon dioxide plus water plus ATP, because we have to get the energy by you can say hydrolyzing ATP in presence of the enzyme pyruvate carboxylase. That will be changing into oxaloacetate plus inorganic phosphate plus ADP. This reaction will be catalyzed by ligases. Remember Krebs cycle, this was one of the important reactions under that particular category. So having discussed the different classes of enzymes as hydrolysases, oxidoreductases, transferases, lyases, isomerases and ligases, let us shift our discussion to characteristics of enzymes. What are the different characters that the enzymes possess? So for their characteristic, we will be telling that first is their proteinaceous nature. This is quite obvious because enzymes are usually globular proteins except the 2 RNA containing enzymes. The one is ribozyme and another one is ribonuclease P. Second one is their efficiency. Enzymes have to be efficient, otherwise they will not work in biological systems, right? So sorry. The efficiency of enzyme depends upon the number of active sites that are present over an enzyme. The number of substrate molecules changed per minute by an enzyme is called as its turnover number. Suppose an enzyme within a minute can change 10 substrate molecules into the corresponding product. So its turnover number is 10. Higher the turnover number, obviously more efficient the enzyme is. The third one is specificity. The enzymes are supposed to be specific. in their action. Different enzymes can act on the same substrate, but they can give rise to different products. Similarly, an enzyme may act on different products to produce different end products. Second, next in line is their chain of, sometimes the enzymes, they catalyze a cascade or chain of reactions. That is also one of their important property. Enzymatic reactions are not isolated. Sometimes they occur in quick succession, or a team of enzymes work together to perform multi-step reactions. Remember, clotting of blood, that will come under this category. Next characteristic will be the colloidal nature of the enzymes. So enzymes are supposed to be hydrophilic and they form hydrosol in free state. Next is their reversibility. It is not the reversibility of the enzymes, but the reactions that they catalyze. Theoretically, almost all enzymatic reactions are reversible. However, the reversibility is dependent upon the energy requirements or sometimes upon the availability of reactants. Also, it is dependent on concentration of the end products and the pH of the reaction medium. Then the last category will be their temperature sensitivity, as most of the enzymes are made up of proteins. So that is why they are heat sensitive or thermolabile, and most of the enzymes work their best within 25 degree to 35 degrees Celsius, which is called as their optimum temperature range. If you increase and decrease the temperature, that is if you get them at freezing temperatures or you can denature them at higher temperatures, they become inactive. So having said this, now let us understand mechanism of enzyme action. How do they work actually? For this, we have to first understand that what is the activation energy barrier. We can draw a simple graph and we can understand it in a more better way. On this axis, let me put the progress of the reaction. And on the other axis, let me designate the free energy of the system. So suppose there is a barrier which is at the middle. So if an enzyme is able to cross this barrier over, then it will surely change into product. So we can write a reaction, that is when an you can say enzyme that reacts upon a substrate, it forms a transient state which is called as the enzyme substrate complex, which readily breaks into enzyme plus product. But this reaction, when enzyme and substrate react with each other, that is a reversible reaction. Means if conditions are conducive, enzyme substrate complex can change into free enzyme and substrate. But this reaction, its rate of reversibility is quite low. So that is why we ignore it. Suppose in the initial state, the substrate must be present at some free energy state. It is not that the substrate will not be having any energy at the beginning of the, you can say, reaction. So suppose the substrate requires this much peak of the energy and then it will change into the product. So this is the amount of energy that you need to activate the substrate so that it changes into the product, right? So this total area can be represented as energy of activation. The substrate was present at this initial energy level. You have to supply energy from outside. It should reach this energy of activation level, and this is the state where it will be present in that enzyme substrate complex state, so that is called as the transition state. At this point, once enzyme substrate complex is formed, it is a downward or downhill reaction where it will change finally into the product, and the enzyme, because it itself doesn't get utilized in the process of reaction, so it will be changing into the product. So when you are not utilizing any enzyme, then energy of activation is this one. But when you are using a particular enzyme, I told you that they lower the energy of activation, so the same product is present at this, but now you will be needing only this much amount of activation energy and it will change into product. So you are decreasing the energy. Mark the difference between the height of the red line and the green line. Obviously green line is comparatively shorter, so that is the energy of activation if it is an enzyme catalyzed reaction. The red graph shows you when enzymes were not utilized, the green graph shows you the position when enzymes were utilized. So this is the final state when the product is formed. So it is simple to understand. If I try to make you this a little bit more elaborate, then every chemical reaction between the molecules involves both bond breaking and bond forming. Changing of one molecule into another will generally involve changing the starting molecule into a highly unstable state. That is, you concentrate on this reaction that I have written at the top. That unstable state is the enzyme substrate complex, and before the reaction can proceed, to reach the state where the bonds can change, the reaction molecules must absorb energy from their surroundings. When the new bonds of the product molecules form, the energy is released as heat and the molecules return to the stable structure with lower energy. Most of the chemical reactions have this energy barrier separating the reactants and the products. Therefore, an external supply of energy is needed in the start of the chemical reaction. This energy is called as the activation energy. So how can we lower the activation energy? According to the graph, activation energy is quite high for non-catalyzed reactions and it is low when it is catalyzed by an enzyme. The activation energy increases the kinetic energy of the system so that the reacting molecules collide with each other at a faster rate, and that brings about forceful collisions between the reactants, and the enzyme lowers the activation energy either by taking the reactants out of the solution state, or they dissolve it, or they establish weak bonds between the reactants and enzymes, bringing reactant molecules close to one another in the region of the active sites and developing strain in the reactant bonds, or the bonds which are present in the reactants, by electrophilic or nucleophilic attack. They make the bonds weaker in the substrate so that they can break into product readily. So enzyme substrate complex is formed when the enzyme binds to the substrate, and this is the point where the substrate is bound on the active site, and this is known as the substrate binding site. There are two models which can explain the formation of the enzyme substrate complex. One is called as the lock and key model and another is called as the induced fit model, which we will discuss in our next series of discussions. Thank you.